count you down from 10, 10, 9, 8, 7, 6, 5, 4, 1, go.
Good evening, everybody, and welcome to the inaugural lecture of Professor Nelesh Govender. Ladies and gentlemen, it's so nice to be standing in the Senate room at the University of the Witwatersrand, but we also recognize all our friends and colleagues who are joining us online from all over the world. But it's lovely to be in a room with people in it again. It almost seems, Nelesh, that we're returning to normality. Now, part of the process tonight is to introduce to you the members of the procession. And my name is Jandrell. I have the honor and privilege of being Deputy Vice Chancellor at this university and have the privilege of introducing so many of our inaugurating professors. Also in the procession tonight, and you'll meet him later on, is Professor Johnny Mahlangu, the head of the School of Pathology, of course, our inaugurating Professor Nelesh Govender, and Professor Lucille Blumberg, who later on will deliver the vote of thanks. I'm looking forward to a really good lecture this evening, so no pressure on our inaugurating professor. But could I call on the head of the School of Pathology, Professor Johnny Mahlangu, to introduce Nelesh to the audience. Johnny.
Good evening, colleagues in person and online. It is an absolute pleasure for me to be here today to see the inauguration of Nelesh Govender from our school. Nelesh is clearly a very accomplished academic, as most of you would know. From his biography, and I'm not going to try and read his extremely illustrious CV. Just to summarize his most important accomplishment, some of which were considered in his promotion to a full professor we are inaugurating today. Professor Govender runs a research group in medical mycology amongst a select few worldwide working in the public health mycology arena. A major theme of his research is HIV-associated fungal diseases. His research activities span lab science, observational epidemiology, cost-effective modeling, as well as clinical trials. And in fact, he'll tell us a bit about some of his work doing clinical trials. This research is aligned to his work as a center head at the National Institute of Communicable Diseases and was partly funded by many grantors. He has been very lucky in that he's been successful in many competitive grants amounting to over 150 million US dollars to date. He has received many accolades, amongst which the Silver Scientific Achievement Award and a medal from the South African Medical Research Council in 2017. He's an honorary professor at the University of Cape Town. And owing to his very close collaboration and partnership overseas, he was appointed a visiting professor at St. George's University of London and an honorary professor at University of Exeter, both in 2021. Nelesh is a fellow of the European Confederation of Medical Mycology. He has served as a co-chair and a member of several World Health Organization expert panels and guidelines. These are expert panels that have developed many guidelines that we're using today in the field of mycology. He was the president of the Federation of Infectious Diseases Societies of Southern Africa until 2021, and is currently co-chair of the National Advanced HIV Task Team and the National Neonatal Sepsis Task Team. He's going to entertain us this evening, and the subject of his talk is Entangled Life and Death, the Global Fungal Epidemics. It is my pleasure to welcome you, Nelesh.
Good evening, everyone. Good evening to the distinguished members of the procession, to my friends, family, and colleagues in the room, and to the various people joining online from across the world. Thank you so much for being here today. It's an absolute privilege to be able to deliver my inaugural lecture in the WITS centenary year. It's been 30 years since I started at WITS as a medical student, and I've spent half my medical career working in medical mycology and in public health. The title of my lecture refers to the high mortality associated with fungal infections. Many of the causative pathogens exist at some stage in their life cycles as tangled filamentous masses. This is really a good opportunity for me to raise public awareness around fungal diseases. And so this public lecture is really pitched at a non-technical audience, but I hope that my colleagues who are experts in medical mycology will also enjoy this lecture. My research work has truly been a collaborative effort. It's been anchored in my excellent team, pictured here, during our World Antibiotic Awareness Week celebrations in November 2021. And the work's been elevated by my collaborators from all over South Africa and across the globe. This is an outline of what I'll be discussing today. I'm going to start by talking about the astonishingly diverse world of fungi. And then I'm going to zoom in to our body as a planet from the perspective of our fungal colonizers. A small proportion of fungi actually are pathogens, and so threaten plants and animals and humans. And I'll discuss what makes a pathogen a bit later on in the lecture. I'll then move on to an intriguing hypothesis proposed about 15 or 20 years ago, which really discusses if fungi were an evolutionary filter for the rise of mammals. We'll then discuss the characteristics of fungal pathogens, and then the bulk of my talk is really related to fungal diseases, and I'll discuss the spectrum of fungal infections, and then focus on life-threatening infections, which are really the focus of my public health work. And I'll talk through 4 separate vignettes to illustrate some of the work we've done. So let's dive in. Fungi belong to a separate kingdom of life, completely distinct from plants and animals and bacteria and archaea. They're among the most ancient branches on the tree of life, and they co-evolved with plants, and in fact were initially considered to be plants. But fungi are eukaryotic, so in fact they're more similar to animals and humans than they are to plants. There's somewhere between 2 to 5 million species of fungi, and the relationships and major groupings within the kingdom fungi are shown here on this family tree, which is really based on genetic data from approximately 1,600 representative species. Fungi are woven through every terrestrial and aquatic ecosystem on Earth. They exist in a wide variety of feeding lifestyles, morphologies, and ecologies, and they in fact comprise the largest living organisms on the planet. Here you see Armillaria, a pathogenic fungus in a state forest in the United States, which is colloquially known as the humongous fungus. It extends underground for more than 10 square kilometers. The mushrooms shown on this picture are just the above ground fruiting structures of the humongous fungus. Fungi exist as filaments and mycorrhizal networks underground entangle forests. These living mycelial threads interconnect with tree roots. fungi actually communicate through their mycelial networks. A recent scientific paper found that spikes of electrical activity from fungal mycelia often clustered into groups that resemble vocabularies of up to 50 words and could actually be similar to human language. Fungi are primarily saprotrophs, so they digest enormous quantities of biological material, mostly rotting plant matter. Many fungi are harnessed for human purposes. They're delicious to eat. This is the black truffle fungus called Tuber melanosporum, and is actually the fruiting body of subterranean mycorrhizal fungi. We know that unicellular or single-celled fungi called yeasts ferment sugary substances into cider, beer, and wine, are essential as raising agents in baking. Mycoremediation is a relatively new concept, which uses fungi to restore balance in the environment by enzymatically degrading soil and water pollutants. And mycofabrication, another new concept, is the process of using fungal mycelium to grow fully formed structures used in products that can replace styrofoam, plastics, and leather. Psilocybin, which is extracted from certain fungi and has a rich tradition in societies and cultures for a very long period of time, is now being tested for drug-resistant depression treatment in a myriad of clinical trials. The artist Leonie Hampton recently premiered a short film which she titled "Our Body is a Planet." In her words, the film challenges the way we think of ourselves as individual, genetically prescribed entities independent from our surroundings. Without fungi and bacteria, our bodies and biosphere would not exist. We are in partnership with the microbial world. Our bodies are made up of more microbial cells than our own human cells. And indeed, scientific studies exploring the human microbiome show that from the tropical jungles of our armpits, feet, and groin, to the oily swamps of our forehead and nose, the arid plains of our forearms, and the dark cavernous recesses of our guts, we host an astonishing variety of microbes, including bacteria, fungi, and viruses. You can see from this picture here that eukarya, indicating fungi, represent a small but non-trivial proportion of our microbial colonizers. So disease-causing fungi or fungal pathogens account for a very, very small proportion of all fungal species, so of the 2 to 5 million fungal species. And yet these pathogens threaten plants and animals that we've domesticated. As an example, Panama disease, which is caused by a fungus called Fusarium oxysporum, threatens banana crops globally, including in Africa. In reaction to these fungal pathogens, our monoculture food crops are treated with fungicides. The backlash, as we'll discuss later, is an emergence of antifungal resistant pathogens. Amphibians now face mass extinction across the globe. Mammals are naturally resistant to fungal infections, and we'll talk about that a bit later again. That's partly related to our elevated body temperature. But amphibians are cold-blooded ectotherms, so they lack the thermal environment, which is exclusionary to pathogenic fungi. Many species of amphibians are under severe pressure from an ancient aquatic flagellate fungus called a chytrid fungus, which colonizes their skin, thickens their skin, and then interrupts gaseous exchange. The spread of these chytrid fungi has been hastened by the global amphibian trade, This is now panzootic, which is the equivalent of a human pandemic, with species declines or extinction events globally. This is the little brown bat. Bats maintain a very high core body temperature when they're active, but when they're hibernating, their core temperature drops precipitously. At this stage, they're susceptible to a fungal infection known as white nose syndrome, and that's caused by a fungus called Geomyces destructans. This has decimated bat populations in North America. We now live in an age of mammals. Mammals are among the most dominant life forms on Earth, not necessarily the most numerous, but certainly the largest terrestrial and aquatic vertebrates. This is also the Anthropocene, in which the human population has exploded and our activities have begun to dominate and shape the world. But this was not always the case. Could fungi explain the dominance of mammals and ultimately humans on Earth? So we know that dinosaurs emerged approximately 220 million years ago. They were the dominant terrestrial vertebrates after the Triassic-Jurassic extinction event. They were possibly feathered, and their modern descendants are birds. Dinosaurs themselves were wiped out after the Cretaceous Tertiary boundary event about 65 million years ago. The current hypothesis is that there was a massive asteroid impact near the Yucatan Peninsula. Photosynthesis shut down for up to 6 months, leading to cooler Earth temperatures for up to a decade, massive deforestation, and a fungal bloom as the Earth became a huge compost heap. There's sufficient numbers or quantities of fungi to leave traces in the fossil record. So could fungi have created an evolutionary filter, allowing mammals to dominate? The dinosaurs which survived the asteroid impact were possibly susceptible to fungal infections, especially because they may not be able to maintain an elevated body temperature with the sun shielded for such a long time. Mammals are smaller, and we're naturally resistant to fungi. It's a combination of two things. We've got both an innate and adaptive immune system, and we're also able to maintain an elevated body temperature. To maintain a core temperature which is higher than our environment is very expensive physiologically, but this, amongst other things, confers intrinsic resistance to most fungal infections, but not to a small group of fungi which actually have learned to grow at our body temperature. These are pathogens. As the Earth warms again with global warming, we expect to see the emergence of thermotolerant fungal species in the environment. And so humans may lose our competitive advantage, especially as we weaken our immune systems with life prolongation measures used in modern medicine. So let's talk a bit about fungal pathogens. Fungal pathogens are generally microscopic in their disease-causing states. Here you see examples of hyphal filaments, pseudohyphae, fungi existing as yeasts or conidia, and encapsulated yeasts, cryptococcal titan cells, or coccidioides spherules. But when they're cultured in the laboratory, one can see their colonies with the naked eye. So this is an example of a fungus, a unicellular fungus or single-celled fungus called Cryptococcus neoformans. Here stained with an India ink, which is a negative stain. You can see that the size of this fungus is approximately 10 microns. A micron is about 1,000th of a millimeter. And of course, these fungal cells can really only be seen under the microscope. But when you grow the same yeast on culture plates, you can see the colonies visibly with the naked eye. And here, Cryptococcus neoformans is growing on Niger seed agar. And so what you can see is brown colonies because of its melanin production. This is an electron microscope picture of a fungus called Blastomyces. And so what you should appreciate here are the tangled mycelia. You can also see the conidia, the round structures, and those are the spores of Blastomyces. Blastomyces is what we call a thermally dimorphic fungus and exists in two forms. When you grow Blastomyces on a culture plate, you can actually see the felt-like mycelium growing on the surface of the agar. So this velvety white colony, which can obviously be seen with the naked eye. The naming convention for fungi is very similar to other life forms. There's a genus and a species name as a Latin binomial. I wanted to make the point here that it's really important to assign as well as to restore indigenous names in taxonomy. My colleague Tsidiso Mapanga, a medical scientist in my team, identified a novel fungus causing an illness called blastomycosis in South Africa as part of her PhD work. We named this fungus Blastomyces mzansi. Mzansi means south in a local South African language. Blastomycosis is an important fungal infection, and in fact, the disease can resemble tuberculosis. So now let's talk about the spectrum of fungal diseases. Fungi cause 4 main forms of human disease, from allergic conditions to superficial infections to disfiguring and finally life-threatening deep infections. The superficial infections involve just the uppermost layers of the skin or the mucous membranes. Here you see a picture of a patient with oropharyngeal thrush or candidiasis. Superficial infections are relatively easy to treat. And so this patient was treated with 4 days of treatment, and you can see that there was a rapid response. Disfiguring infections are usually caused by traumatic implantation of organic matter, so usually soil or plant matter, contaminated with fungal pathogens into the deeper layers of the skin or into the eye. These are neglected tropical diseases which tend to affect the most impoverished communities in the world. And these are very difficult infections to treat. They often require months of antifungal treatment and often require surgery. Deep or systemic infections require treatment in hospital and are associated with very high mortality. They occur both in high income and low income countries, and particularly occur among people with weak immune systems. This is really the focus of my public health work. And so I'm going to now discuss 4 short vignettes illustrating some of the work we do in fungal infections. So we're going to start with a condition called cryptococcal meningitis. The fungal pathogen Cryptococcus neoformans or Cryptococcus gattii is a yeast. So it's a single-celled fungal organism. It causes a life-threatening brain infection or meningitis in people, usually with advanced HIV disease. This yeast has a large polysaccharide capsule, so this large gelatinous capsule. And this, in fact, is its major virulence factor. It allows Cryptococcus to hide from the immune system, for example. But this is also the fungus's Achilles heel, because unique components of the fungal capsule, so-called antigens, can actually be accurately and rapidly detected by dipstick tests. And these tests have actually opened up the possibility of large-scale screening programs for crypto disease, with the potential to save many thousands of lives. The screen and treat intervention is a relatively simple concept. The first is to identify people at high risk of cryptococcal disease. And as I said, people with advanced HIV disease, usually with a CD4 count below 200, are at highest risk of crypto disease. The next step is to screen their blood for early crypto using the dipstick assay. And if the test is positive, to treat patients with an antifungal medicine called fluconazole. And if you've managed to identify these patients before they've developed meningitis, there's a distinct possibility to prevent meningitis and thus prevent deaths. Screen and treat for crypto disease is now being implemented globally. It's a recommendation that's in place from the World Health Organization and has been so for the past decade. Guidelines in 27 countries across the world have included screen and treat, and South Africa has been a world leader. We've initiated the largest screening program in the world. So between February 2017 and September 2021, more than 1 million people with a CD4 count of less than 100, so these are people with advanced HIV disease with a CD4 count below 100, were automatically screened for cryptococcal disease in public hospitals and primary care clinics. And during this screening period, more than 64,000 people with cryptococcal antigenemia were detected or diagnosed. So if screening programs are not in place, or screening programs are in place but don't work, cryptococcal meningitis remains a common AIDS-related illness. We estimated that approximately 220,000 cases of cryptomeningitis and more than 180,000 deaths related to cryptomeningitis occurred globally, with approximately 3/4 of the cases and deaths occurring in sub-Saharan Africa. Cryptomeningitis is estimated to be responsible for 15% of AIDS deaths, second only to tuberculosis. So how do you treat cryptomeningitis optimally? The multi-country ACTA trial, which was conducted in several countries in Africa, showed that an old, off-patent medicine called flucytosine was an essential part of treatment for cryptomeningitis. On this slide, you see survival curves from the ACTA trial. Each of these colored lines represents a group of people treated with different antifungal medicines. The purple line represents the group treated with amphotericin B plus flucytosine, followed by another medicine called fluconazole. And this group had the best survival at 10 weeks. This particular regimen requires that patients be treated in hospital at least for a week with intravenous treatment, and then they can be stepped down to oral medicine. This is the current World Health Organization standard treatment for cryptomeningitis. Unfortunately, flucytosine is not widely available. This is partly because of limited manufacturing, and until recently because of very high costs of this medicine. It was also not registered in South Africa until December 2021 by the South African Health Products Regulatory Authority. However, since 2018, we've been using flucytosine through an access program in South Africa. And so confirming the results of the ACTA clinical trial, we've shown through the access program that mortality was halved if flucytosine was used in the treatment of cryptococcal meningitis in routine care. So clearly, flucytosine works in routine care, adding further impetus to make sure that this essential medicine is widely available. A new clinical trial called the Ambition trial has recently been published and has simplified cryptomeningitis treatment by incorporating a single intravenous dose of liposomal amphotericin B along with flucytosine and fluconazole. As you can see from the slide, the graph on the upper right-hand side, although this regimen was not any better than the current standard in terms of survival, it was shown to be safer for patients and was also preferred by healthcare workers because it's much easier to administer. So we now have the tools to drastically reduce mortality from cryptomeningitis. Firstly, screening programs have the potential to completely prevent cryptomeningitis. But if cryptomeningitis does occur, the first and most important intervention, of course, is to diagnose the condition, because undiagnosed cryptomeningitis is always fatal. So diagnosis plus fluconazole improves survival marginally. Appropriate antiretroviral timing and ensuring that patients have spinal taps during their hospital admission improves their survival even further. Adding flucytosine to fluconazole improves their survival even more. Using the World Health Organization standard improves survival to 70%. And the recent AMBITION trial has shown us that we can now implement treatment safely and even more easily. But in low and middle income countries, these interventions are seldom being used universally. And so there's a huge amount of implementation work that still needs to be done. We're now going to turn to another condition that occurs among people living with HIV. And this is a condition called emergomycosis. In South Africa, emergomycosis is caused by a fungus called Emergomyces africanus that we only recently discovered. So I'm going to discuss a patient with advanced HIV disease who over several weeks developed a rash, which started on his face and then spread to his limbs and to his trunk. These lesions were initially macular, they became papular, and then eventually became nodules, which ulcerated and crusted. The patient also had fever and lung involvement. We grew a fungus from the patient's blood. So this was clearly a deep infection, not just a superficial fungal infection. Emergomyces africanus is a special type of fungus that grows in two different forms, depending on the temperature. It's a mold or a filamentous fungus at room temperature. It's shown here on agar plates. And you can see that after 6 weeks of incubation, you see these wrinkled brain-like colonies growing on the surface of the agar. Here's the scanning electron microscope showing you the tangled hyphae as well as the conidia or the spores. And here is Emergomyces africanus growing at 35 degrees Celsius, so equivalent to human body temperature. And at this temperature, the fungus converts to the yeast form, the unicellular form. So in fact, it really is specially adapted to cause human infection. This is what it would look like under the microscope when stained with a special stain called the Gram stain. This patient responded very well to intravenous antifungal medicines in hospital. His lesions healed very well, although with some residual scarring. However, the outcome is not always this good. In a case series of patients with emergomycosis, almost half of the patients died, partly because diagnosis was delayed. So this patient's infection was really caused by Emergomyces africanus, which until recently had not been described. It's called Emergomyces because of the apparent emergence of these fungi around the world, although the emergence is more a consequence of improved diagnostic methods rather than a recent emergence. Within the genus Emergomyces, there are 5 species, and cases have been diagnosed on 5 continents. Emergomyces posteriorus in Europe, Asia, and Africa, Emergomyces canadensis in North America, Emergomyces europaeus from Europe, Emergomyces orientalis from Asia, and with the most cases, Emergomyces africanus, only from Southern Africa to date. We're going to turn now to another condition called candidemia. So fungi in the genus Candida cause a bloodstream infection called candidemia. This is usually diagnosed in people admitted to hospital, and many thousands of cases occur annually in South Africa and across the world. The attributable mortality associated with candidemia is around 30%, even with treatment. Over the last decade, a newly identified species called Candida auris emerged as a cause of candidemia. In a 2019 report from the United States Centers for Disease Control and Prevention, which really tried to rank 18 antibiotic-resistant germs, Candida auris was listed among the top 5 most urgent threats. Antifungal resistance is often a consequence of interconnected ecosystems. We often think about antibiotic resistance using a concept known as One Health. So factors such as increasing use of chemicals in agriculture, increasing numbers of people at risk of fungal infections, a change in climate, changing microorganisms in the environment, as well as the changing virulence of such microorganisms are drivers of resistance and lead to a vicious cycle where pathogenic fungi are exposed to fungicides in the environment which mimic human antifungal medicines. These fungi then develop resistance, and resistance is potentially amplified in environmental hotspots. In the case of a fungus called Aspergillus, often in compost heaps. And then people in high risk or who are at high risk for these fungal infections, mostly in healthcare environments, can then be exposed to drug-resistant fungi. Over the last decade, we tracked the near simultaneous, apparently independent emergence of 5 lineages of Candida auris causing human infections with its subsequent spread to 6 continents. Based on genomic analyses, we found that Candida auris had emerged probably in the last 350 years, with the drug-resistant strains probably emerging in the 1980s, which is around the time when we started using antifungal medicines called the azoles. And so, these fungi emerge simultaneously. These drug-resistant strains emerge simultaneously. A year ago, Candida auris was reported from the environment from the very first time. Candida auris was cultured from a pristine salt marsh and a tourist sandy beach in the Andaman Islands in the Indian Ocean. One hypothesis is that Candida auris began life as a tropical wetland saprotroph and then adapted to high salt and high temperatures there. Even more recently, Candida auris has been found on the surface of stored apples, suggesting a possible amplification step for antifungal resistance en route to the hospital environment. In South Africa, we've conducted national surveillance for candidemia 5 years ago, and we found that Candida auris was a major healthcare-associated pathogen in South Africa, accounting for more than 1 in 10 cases of candidemia. The epicenter was in Gauteng province, and Johannesburg and Pretoria are located in Gauteng province, just to orientate you. Over the last 5 years, we've seen a further spread of Candida auris to other South African provinces, and of particular concern we've seen large infection outbreaks in newborn baby units, which we hadn't seen 5 years ago. So finally, I'm going to discuss a disease called sporotrichosis, a disease which is really entangled with the history of the Witwatersrand. In the early 20th century, enormous outbreaks occurred on the gold mines. Contaminated timber props were thought to be the source of the fungus, Sporothrix schenkii, which was then inoculated into the miner's skin, causing the classical lymphocutaneous lesions. In the 21st century, we've seen far fewer outbreaks, except where traditional mining techniques are used, and also more recently where illegal mining activities occur. About 10 years ago, we investigated an outbreak on a gold mine near South Africa's border with Eswatini. A group of miners had Sporothrix schenkii cultured from their skin lesions, and we conducted an outbreak investigation. After a harrowing trip into the very depths of the mine, we found evidence of Sporothrix in the environment too, although it turned out to be a completely different species called Sporothrix mexicana. And so from a genomic point of view, we weren't able to close the loop and work out the environmental source for this outbreak. I'm going to end my talk by briefly talking about 3 new projects that I'm going to be involved in over the next 5 years. The first is the EFFECT clinical trial to determine if a combination of flucytosine and fluconazole works better than fluconazole alone for early crypto diagnosed through screening. We will involve 11 sites in South Africa and Tanzania. We plan to enroll 600 people into the trial over the next 3 years. And we've involved multiple partner institutions in South Africa, in Tanzania, and in the United Kingdom. The investigator team is pictured here at our March 2022 launch investigator meeting. So a very exciting trial, which I hope will provide some answers in terms of the best possible treatment for cryptoantigenemia. Secondly, Professor Joe Jarvis from the London School of Hygiene and Tropical Medicine and I will co-lead a new global health research group on HIV fungal infections. This is the first group funded by the National Institute for Health Research for fungal infections. And we have an ambitious program of work tackling several deadly HIV-related fungal infections: cryptococcal meningitis, histoplasmosis, emergomycosis, pneumocystis pneumonia, and tularemycosis. We've got partners in 7 countries, and these are some of the investigators, again, in a recent investigator meeting. And finally, in partnership with the United States CDC, we are building a regional network for fungal genomic surveillance, including pathogens such as Candida auris. Genomic surveillance, of course, is the process of collecting samples containing pathogens, analyzing their DNA, and looking for similarities and differences. We will focus on fungal genomic data and use this information to monitor and control fungal diseases in the region. So fungal infections, in conclusion, will continue to challenge us in clinical medicine and public health. Over the last 2 years, at least, we've seen emerging populations at risk. Severe COVID, for instance, is an important risk factor for systemic fungal infections such as mucormycosis, COVID-associated aspergillosis, and various other infections, including candidemia. We also expect to see emergence of other fungal pathogens, all related to our changing environment. As scientists, clinicians, and public health workers, our role is really to understand these pathogens, understand our host immune responses to them, and then optimize the diagnostics, the treatment, and prevention strategies. And most importantly, to do all of this without neglecting people who live in places with the fewest resources. So this lecture is dedicated to my paternal grandmother and both my parents. And I'm going to end on this family picture taken about a week ago. Thank you.
Professor Nelesh Govender, you can indeed profess. Congratulations. But to formally thank you, I'd like to call up Professor Lucille Blumberg to propose the vote of thanks. Lucille.
So congratulations, Nelesh. You really delivered a masterpiece from the magical world of medical mushrooms and molds. You've taken mycology from what was a neglected subject, very much the orphan of microbiology, and grown it into what it is today in your department and elsewhere. You've embraced the epidemiology, the clinical side, prevention and treatment, the teaching and training. It's truly remarkable. Really want to highlight your work in HIV-related mycoses, especially the cryptococcal program of test and treat, which I think changed the face of how we manage cryptococcus in our AIDS pandemic. And I think the ambition now for 0 deaths from cryptococcal meningitis is an achievable one. So I think that has to be one of the highlights of your career. But there's no stopping you. And I think we heard of 4 very ambitious programs that are going to keep you very, very busy in the next few years. So congratulations on this remarkable achievement. We look forward to many, many trials and not too many tribulations around fungi in the next few years. And I'm sure there's going to be some new emerging fungi that are going to come out of your laboratory and your research projects. Behind every budding mycologist, there are some really important people. So I'd like to acknowledge your mum who's online, couldn't be here today. I'd like to acknowledge Paul, your sister and your niece who've joined us this evening, and many colleagues who've worked with you over many years. So I'm very proud of you tonight, Nelesh, and looking forward to many, many wonderful mycological miracles in the next few years. Congratulations, and so well done.
Thank you very much. So, friends and colleagues, this formally brings us to the end of the proceedings this evening. I just want to make one observation, and I think it's such an important one, which both Nelesh and Lucille highlighted, and that is that any endeavor of excellence involves people, and to a very, very large extent, they involve your family. So let's not forget the role that our families play and how important they are in terms of every step that we take as individuals, but actually as members of families and communities of scientists. So thank you all very, very much for being with us. We really appreciate it. And once again, Professor Nelesh Govender, congratulations. Thank you so much. Good night, everybody. That was wonderful. Great. Thank you. And speaking of the engineer, they want to ask.